So my name is Ruthie. Ruth Shouval is my full name. I'm a postdoc at the Lindquist Lab, in which we're sitting right now, and this is in the Whitehead Institute, which is an independent research institute that's affiliated to MIT. We study cancer, we study neurodegenerative diseases such as Alzheimer's and Parkinson, and we study prion diseases, which are the mad cow disease. I look at the interaction between the cancer cells and their environment, so the other normal cells that are around them, and how the cancer cells interact with these normal cells and recruit them to help the tumor grow and progress instead of trying to kill the tumor, which is what normal cells would want to do. I was born in Israel and born and raised in Israel. I think I always knew I wanted to be a scientist. My parents are both scientists, and so I was sort of born into this. And I did my PhD at the Weizmann Institute in Israel, and then I came here to do my postdoc. And the Whitehead Institute is an amazing place to do research in, and you can collaborate with both clinicians and scientists, and all of them are very close and very reachable. Here are the benches of everyone, and back here we have our robot room. I work on a transcription factor, so that's a protein that regulates the expression of other proteins in the cell. And we found that this transcription factor, that is called heat shock factor 1, is important for the progression of cancer. So we look at breast cancer and we look at it in patients. We collaborate with physicians to look at the relevance of what we find here in the lab with patients. This room is at 37 degrees at all times, and this is where we grow bacteria. So in all these flasks we have yeast growing, and so we put just a little bit in that flask with media that the yeast likes to grow in, and then overnight we get a full culture of grown yeast. This is where we have all our robots who do some of the stuff for us. So as you can see, this plate now that I filled could be, instead of me filling it, it could be filled by this robot if I would place it here. Or now that I already have filled it, I can use this robot right here to analyze the wells that I filled. So what I do is I place this plate right here, but then here there is some signal. And in the same way that I now did one plate, I could do up to fill this whole stack with plates, so do like 24 plates, put them here, go out for lunch, come back later and pick it up. We have microscopes that are extraordinary and can help us get into very good resolution of looking at our biological samples through time and getting through a lot of samples very quickly. A lot of people think that scientists just, work, that it's a very lonely process, that they work alone at their bench and they don't really interact with people, they don't really talk to other people. And I think that is very misleading. I think that a lot of our work here is a collaborative work, and we work as a team, and we each have our independent project, but we work as a team, which I think is fantastic. I think there are very few jobs where you're paid to just be creative and think of whatever you want and then actually try and do that. You don't necessarily have to be in Boston and in Cambridge to do great research. I think being creative and being able to find the right question. There are minutes of frustration. There are many periods of frustration here, but finding a clue or getting a good result after you tried to find it for a long time and then finally finding it is an amazing feeling. It's just amazing. And then thinking that it might actually help people and help sick people, what could be better than that?